Hello, we are Luis, Nadia, Miguel, and Magdalena, and we are going to explain how to plan a relational study of the effect of tattoo ink in DNA methylation and gene expression.
Epigenetics is the study of heritable changes in gene expression that do not involve changes in the underlying DNA sequences but are dependent on the environment. DNA methylation is among the most studied epigenetic changes, and it is the covalent addition of a methyl group on a cytosine residue. It plays a critical role in long-lasting programming of gene expression by affecting the interaction with DNA of chromatin proteins and specific transcription factors. Therefore, DNA methylation controls gene activity. For instance, methylation of promoters often leads to repression of transcriptions and thus of gene expression. This repression can be due to direct effects of methylation on transcription factors or caused indirectly by binding of repressor proteins to methylated DNA. The study of DNA methylation has become very relevant as it has been causally related to several diseases, including monogenic and complex ones, such as cancer due to hypermethylation in tumor cells. Following the steps of studies focused on the effect of environmental factors on gene expression, we have decided to study the possible DNA methylation effects of tattoo ink in humans. We must use a controls versus cases approach. The two techniques chosen to carry out this project were a microarray chip to assess DNA methylation and RNA-seq for a gene expression analysis. Results obtained from each group could then be compared in order to look for significant differences. Finally, so as to study the relationship between methylation and expression, we would have to use the Pearson's coefficient to calculate correlation. The chosen sample size was of 300 volunteers that provided informed consent, 150 controls and 150 cases. As we are just looking for broad changes in the methylome as a whole, this sample size will be enough. It allows to identify small effects with an 80% power. If we were looking for something more specific, a larger sample could be required. To ensure association of methylation with the tattoo ink, we must reduce variability among the samples as much as possible, defining the patient's profile in both cases and controls, such as age and certain habits. We have limited our sample to people aging from 25 to 60 years old and non-smokers to avoid interference in the study. It has been also decided to study possible gender-specific effects on differential DNA methylation. Therefore, we have looked for similar number of men and women in cases and controls, so as to assess possible sex interaction. With that objective in mind, men and women in both cases and controls were analyzed independently so as to see if there was a significant difference in methylated CpGs among them. As tattoos are an easily selectable phenotype, we have looked for cases with tattoos exceeding a minimal amount of ink per individual, addition of all tattoos in cases they have more than one. The chosen minimum size was 80 cm square. A time restriction was also added so that it were more likely that cumulative methylation changes occur. Therefore, tattoos must have been done at least 1 year before the study. Although there are short-time natural changes in the methylome, a relation with methylation and time of exposure and dose has been observed in several researches. On the other hand, control's profile must be the same than the one of cases, but with no tattoos. Therefore, they must be non-smoker people among 25 and 60 years old. Regarding sample extraction, the best option was to obtain the DNA and RNA from whole blood samples, as it is an easily accessible material and the most innocuous way. The procedure for its obtention is just the same than in donations. This was carried out by professionals and following all health precautions. Once we obtain participants' whole blood samples, the next step is the purification of DNA and messenger RNA in order to assess methylation and to evaluate gene expression through RNA-seq respectively. Genomic DNA for DNA methylation analysis was extracted from cell lysate using the GeneElute DNA mini-prep kit. Then DNA concentrations were determined using the nanodrop. Total RNA was extracted from cell lysates using the RNA-easy mini-kit according manufacturer's protocol and treated with 6 units DNase-1. RNA quality and integrity was then assessed for all samples on the Agilent bioanalyzer. Samples with low quality that do not reach the established threshold could be removed. Then poly-A-containing mRNA was purified using poly-T oligo-attached magnetic beads. Finally, RNA concentrations were also determined with nanodrop.
The methylation technology we decided to use was the Illumina 450K methylation chip, an array that evaluates the methylation state of 450,000 CpGs, which cover almost 99% of ref-seq human genes. So it was the first approach to interrogate widely the cell methylation patterns. That's why we did not use bisulfite sequencing, since it would cost us much more time, money, and work for data obtention and its processing. This technology needs, first of all, a bisulfite conversion, which is a chemical conversion of DNA whereby applying sodium bisulfite to the DNA samples, the non-methylated cytosines turn to uracil, and methylated ones remain equal. Then samples are fragmented and hybridized with the probes. These probes stop just before the possible methylated cytosine, and then a single base extension is performed. So if the CpG was methylated and the signal would be the one of the C or G fluorophore, and if it wasn't, it would give the signal of A or T fluorophore. This extension reaction is observed by the Illumina HiScan SQ scanner, and it gives us two values for every position. Beta value corresponds to the percentage of methylated cytosines, and M value to the logarithm to the base 2 of the ratio of intensities between methylated and unmethylated probes. We performed our following experiment just with the M values given by the Illumina. Once the M values are isolated, we have to deal with many data. So our first approach is to remove all the data that can lead us to some error. Firstly, we deleted data that did not achieve a high-quality signal, maybe because hybridization wasn't ideal, or maybe just the optic signal wasn't enough to determine a valid result. Secondly, we removed CpGs that were closer to 20 base pairs to polymorphic fragments, because hybridization could have been altered. Since beta values readouts has the total intensity in the denominator, it is a robust value, but working with the M values is more delicate. So that's why once these filters were applied to our data, we used an R script that follows quantile normalization method in order to obtain more equally distributed data and remove background noise. Then we started the statistical association analysis by a multivariable linear regression method, in which the M value was the variable to explain, and the variables that explained the methylation signals were sex, age, and tobacco. For that, another R script was used to perform the methylation status calling procedure. This method studies separately the methylation status of each CpG. The script takes also into account the false discovery rate, that applies a more stringent statistical threshold to count a CpG as methylated. Then, finally, we considered a specific site as differentially methylated if it had a higher difference than 10% in the methylation signals between cases and controls, and a lower p-value than 0.05.
For the analysis of gene expression in both study groups, we have decided to use RNA sequencing with NGS instead of expression microarrays or qPCR, since it's the most used strategy nowadays due to its main advantages. As a matter of fact, sequencing is becoming cheaper and cheaper every day, which may explain why it is now the predominant choice. As it can be observed, in 2016, RNA sequencing already surpassed the microarray usage. The procedure has two main steps. Firstly, the sample preparation and generation of the library, sequencing, of course, which will be performed by an external company, and secondly, the data analysis, which involves mapping the rates, normalization, quantification, and visualization. For the library construction, Illumina TrueSeq RNA Library Prep Kit V2 will be employed, which allows multiplexing by means of its 24 unique indexes. Previously purified mRNA must be retrotranscribed into cDNA for the construction of the cDNA libraries. The library will be non-stranded, since the aim of our experiment is to analyze wide and unlocalized methylation profiles, and we are not interested in knowing whether specific DNA regions are methylated or not. Then, the library will be sent to Illumina to sequence by HiSeq Illumina 2000, and we will obtain single-end reads of 100 base pairs. Base calling will be performed by the offline base caller Illumina V194. Well, we will be given a FASTQ file with the reads and an associated quality per base. Once we're given the sequencing results, we will need to perform a series of downstream analysis. Firstly, we should assess the quality of the sequencing in order to remove those reads whose quality could compromise the following steps. Catalog version 1.1 will be employed to eliminate those reads which may contain adapters in the sequence, and the platform SecKit will be employed for the manipulation of the FASTQ files resulting from sequencing, hence performing the actions related to filtering and trimming. The next step will be to perform the mapping of the remaining reads against the human reference genome. For such purpose, TopHat will be employed, a free source available online and a splicing over algorithm which does not rely on known splicing sites. Afterwards, we will proceed to quantify the expression of the genes, taking into account the number of aligned reads to each genes, for which the HTSec count tool of HTSec, a framework that uses Python program, will be used. This script is specifically designed for differential expression experiments, so only read mapping unambiguously to regions will be counted, and not considering those maps to multiple positions or overlap to genes. It would be considered that the higher number of reads are aligned to each gene, the higher it is expression level. So, should we start assessing differential expression? No. First, a normalization procedure is needed so that we can compare between samples. Some biases exist, such as sequencing depth, gene length, GC content, and RNA composition, of which sequencing depth and gene length are especially important. Normalization by RPKM will be carried out, since it is one of the most basic and widely employed, even though it does not consider the RNA composition bias. The RPKM normalization will be carried out by executing a script designed by us in Python. As input for the script, we should introduce the file containing the count matrix generated by HTSecCount. The normalization for every gene of every sample should follow the indicated formula. For the differential expression analysis, we will make use of HR. From the data, the quasi-likelihood dispersions will be estimated. Afterwards, we will check for significant differential expressed genes by using the QLF test, from which a p-value and a false discovery rate values will be obtained for each gene and ranked. A multiple test correction should be applied in order to minimize the errors and false positives resulting from analyzing such a high amount of genes. We will get adjusted p-values, one for each gene. We should eliminate for further studies those genes above a certain p-value threshold and a certain FDR threshold. The remaining genes, which fulfill both conditions, will be the ones we consider differentially expressed. Finally, in order to observe more intuitively the differential expressed genes, we can generate the display plot. The last step with the differential expression procedure will be to study functional enrichment. That is, to analyze if there is any biological pathway whose expression has been altered specifically. For such purpose, we will cluster the differentially expressed genes according to Go terms, identifying the cellular function or pathway. We will make use of the GOANA function of HR, which uses the NCVI RefSec annotation. By applying the top-Go function, the 10 most enriched Go terms will be viewed. We will analyze if the Go terms of the case group are distinct to those of the control group, and if any biological pathway or physiological activity is enriched.
So then, we must link the data obtained from the methylation and expression analysis of each individual to identify a potential relationship between these two factors. Correlation tests could be performed overlapping the expression data and the beta methylation values previously normalized. Pearson's coefficient could be calculated by using R, and this coefficient could provide the desired information about the methylation expression relationship in such a way that it could be possible to answer the following questions. Is there a linear relationship between this kind of data? And if there is a linear correlation, does methylation lead to repression or overexpression of the associated gene? Pearson's coefficient could be a number between -1 and 1, meaning the following. If Pearson's coefficient equals 0, there is no linear correlation between the given information. But if Pearson's coefficient is equal to 1 or -1, a complete linear correlation exists between the given information. Now, it is important to keep in mind that this kind of analysis are very sensitive to outliers. So it could be useful to plot the methylation data against the expression data in a scatter plot in order to have more information in the analysis. In addition to this, it could be necessary to perform a multiple test correction of correlation p-values with R to check the statistical significance of this analysis. As you know, budget is an important issue in the planification of a project. So here you can see in the table the cost of the resources that we could use in our experiments. Finally, we estimated that we could spend around 200,000 euros. Finally, the time spent in this experiment was estimated by keeping in mind the different phases. In sample extraction and preparation, at least 1 week would be needed. On the other hand, the methylation analysis could be shorter. Only 60 to 90 minutes per chip are needed for the reaction with bisulfides and elongation. Then, each array could be analyzed by the high-scan scanner, and this process could last around 11 to 29 hours. Keeping in mind the shipping time, 4 to 5 days could be enough for this step. Next, RNA-seq could take 1 week approximately. Library preparation could last 16 hours, while sequencing, data analysis, and processing could last a maximum of 4 days. Finally, the analysis of the results and the article writing could be the longest step, taking even several weeks.